Hi, good morning. So it's good to be back here. It's been 2 years since the last primer. The last primer talk I gave was definitely not one of the most awesome ones because it was...
No, that's why you were 2 for 3, I think so.
2 for 3. The last one was November 9th, 2016, which was also the day after an election. And I've not made the same mistake I made then because I have not looked at the news. So I saw a newspaper out, and I saw it folded over and said, "History made." I'm like, history was not made, not until 10 a.m. So I really have no idea what's going on in the world, but I know something about topic models. So for this talk, I want you to consider an artificial data set that I've made. It's very simple. It basically consists of different workouts of various people. So this is a cartoon data set. It's very simple. It's for pedagogical purposes, but it's somewhat realistic. So for example, you have a person 1 who seems to do some weightlifting exercises with barbells. And person 2 seems to be a runner. Person 3 does yoga. Person 4 does some body weight-based calisthenic exercises. We have person 5 who's a runner. And I want you to imagine the ... dots means you have a lot of people like those. So you have a lot of people who do basically some set of weight exercises and some selection of running and some selection of yoga exercises. Then you have some people, like person 1000, who might draw from more than one category. As you can see, that person does a bit of weights and a bit of yoga. And then if you go ... further, you have more anomalous people who can't make up their minds at all. So person 2000 is an example of someone who just does a buffet of all sorts of different styles of exercise and apparently hopes to get fit that way. And we are going to ask and answer a few questions from this cartoon data set. And they're both questions that are really obvious to us a priori, so we're not really doing any high-powered machine learning here because you could have learned it with your own brains, but forgive me for that. So the first question is just, let's discover some different categories of exercise. Now, you know a priori the answers to this. You know that person 1 is doing weight exercises and things like a squat and a deadlift and a bench press and anything with a barbell just belong in a category. But let's pretend that you don't have any prior information. So as far as this data set is concerned, bench press is just encoded as a 1. And downward dog pose is encoded as a 2, etc. So you have no lexical or prior information about what these mean. You're just trying to make clusters out of them. And if bench press is a 1 and downward dog is a 2, etc., it's not as obvious a priori that person 1000 is drawing from one category, which is, as we know, the category of weights, and another category, which is the category of yoga. But we'll discover that. And another question we could ask, again, it's obvious to you, is how do we know that 2 people are doing similar things or different things? I mean, how do we compare different people? So as far as just bench press is 1, downward dog is 2 is concerned, the 2 runners in this data set look totally different because the exercises they do don't overlap. So if you were to represent them as these big binary vectors of 1s and 0s, like person 1's exercise is 1, 0, 0, 0, 0, 0, and then a 1 for whatever component of your vector is shoulder press. If you did that sort of encoding, then the 2 runners, persons 2 and 5, would not overlap. They'd be totally orthogonal. So the question is, how do we discover the obvious and be able to say, no, they're actually similar? So I'm going to do something presumptuous, and I'm going to tell you how you would reason. So I think it's safe to say that the categories we would discover from this would be things like weight training and yoga and running. And the way I think you would reason that is you would look for exercises that occur in the same workout repeatedly. So if you looked at tens of thousands of these workouts, you would probably find that bench press and squat, actually, do people know what squat is? Did you put a barbell behind your head?
You should really demonstrate. I know you do.
Okay, I do. Okay, a squat, you put a barbell behind your back and you go like this. Some people only go this far, some people go this far, and then you you to get up. Okay. Deadlift is you hold a big barbell, you go like this. You start out, you activate it from a little bit from the quadriceps, then your glutes take over and you lift up. Okay. The point is they have a big heavy weight and you lift them. John, that was totally unnecessary. Anyway, what you're assuming is that those things that I just showed occur in the same workout. Not every time, but often enough. And similar for the yoga, which I'm not going to demonstrate, you would see a lot of workouts that have a crow pose and have a dog pose. And you would know that person 1000 is drawing from two categories because you wouldn't see a whole lot of workouts that have both a bench press and a downward dog pose. You might see some, but not enough to really conclude that those belong to a single coherent category. Now, if you're reasoning that way, you're making one more assumption, and it's critical to understand that you're making it. If you're assuming that similar exercises that belong to one category co-occur in one workout, then basically by the pigeonhole principle, you have to be assuming that most workouts don't use a lot of categories. So people like person 2000, who just can't make up his mind at all and is drawing every exercise he has is from a different type of exercise, those people are rare. Most people will say, okay, this is a weights workout, or a weights workout with a little bit of stretching, something like that. So I've put it in a box because this assumption is basically one of the pillars of topic modeling. And we call this a sparsity assumption because we're saying every workout is sparse in that it uses a small number of categories. This is critical. So the question is it sufficient? If you only have this principle that I put in a box, that most workouts draw from a small number of categories, is that enough? And I'm going to argue with this comic that the answer is no. Because if you have a model and what it's qualitatively trying to do is make everyone's workout use a small number of categories, you can satisfy that model and make it extremely happy by putting every exercise into one enormous category, in this case called the category of expending ATP. And that way, everyone's workout would be an ATP expending workout, and you would satisfy sparsity really, really well. But obviously, that's absurd. You haven't learned anything. So it must mean that we need a second qualitative pillar to build a topic model. And the simplest thing to do is if you find the first principle of workouts using few categories, if you find that first principle leads you to a bad conclusion, then the easiest thing to do is make a second principle that fights that conclusion by fiat. So our second principle is that a category of exercise doesn't have a lot of exercise. We'll have some sort of prior belief that you don't just want a big category that gloms every weightlifting exercise and every yoga exercise into one big category. So that way, you do have sparsity. You do try to make clusters of categories, but you don't try to make them too enormous. And basically, these are the reasons topic modeling works. And this is topic modeling. So now that we have the qualitative foundation, let's build a topic model. So what I'm going to build is called latent Dirichlet allocation. We'll get to why it's called that. The abbreviation is LDA, so I'll be referring it to as LDA for the rest of the talk. And it's so popular that it's basically synonymous with topic modeling. That's not exactly accurate, but most of the topic modeling you'll read about is based on LDA, or is just literally LDA with no modification. So what we'll do, it's a hierarchical graphical model. We'll build it from the inside out. We'll start with a really simple model, and then we will add layers of an onion until we're at LDA, and it won't take very long. So the first model that I'll present, I'll call a dubious weight training coach. You could think of this as an app that you put on your phone, and you probably don't sell it because I don't think anyone would buy it, but you give this app to the public, and it tells them, here's a workout you can do. So it has one parameter, which is phi, And that parameter phi says what are the probabilities of any given exercise. So the way this app works is it generates a workout which has, let's say, 10 exercises. For the first one, you roll a die. Second one, you roll a die. Every exercise you choose is random, and it's chosen according to the probabilities in the parameter phi. So for example, if we have a weight training workout, phi's component for bench press would be like 0.1. For a deadlift, it would be 0.2 or something. And it would be 0 for most things. That's our sparsity assumption. So for anything that's not related to weightlifting, you would have a 0. And that's this exercise. You flip a lot of coins or roll a lot of die. So we'll add one layer to this model. And now we'll say, well, maybe our app wasn't very popular because it only gave you weightlifting exercises. It was pretty boring, and some people might want to do other things with their life. So now let's make an app we'll call the dubious CrossFit coach. We still can't sell it for a profit, but we're getting there. And the dubious CrossFit coach has another parameter, theta. And what theta is a parameter that selects different categories of exercise. So if you might have a workout that's mainly weightlifting, and theta would have a component of 0.5 for weightlifting. And it might have a little bit of yoga mixed in, so theta subscript yoga would be 0.1, something like that. And they would add up to 1. And the way you would design a workout with this app would be for the first exercise in the workout, you roll your weighted die according to theta, and then it might show that you get a weightlifting exercise. So then if you choose that category, then you select which weightlifting exercise from the parameter phi. So you choose the category from theta, then you choose from the phi appropriate to that category which exercise you do, and then you repeat. So there's no real sequential logic or coherence to this exercise. There's no correlation between if your fourth exercise is yoga, then your fifth is also predisposed to being yoga again. It's just totally random. You could call it a bag of exercises. So once we're there, we basically have LDA. All we have to do is take those proprietary parameters, phi and theta, that give you the optimal balance of weights and yoga and running, and say, well, actually, we're not so smart. Those are random variables that are discovered from the data. And they're not just any random variables. They're random variables that encode the two qualitative assumptions that we discovered earlier. So theta, the parameter which says which different types of exercise make up a workout, we wanted that to be sparse. So we'll give it a sparse prior. Similarly, and we'll talk about what that prior is. Similarly, we said every category shouldn't use too many different exercises. So for every vector within phi, we will also draw that from a sparse prior that says this phi doesn't put weight to too many different exercises. And we have parameters alpha and beta, which for now we can say they control how sparse that is. So the way it works is that if alpha and beta are really big, it's not a sparse model. And then as they get smaller and smaller, it gets more and more sparse. So if you decrease alpha, it says that you'll have workouts that are predominantly yoga and not much else. And if you increase alpha, you might have more workouts that are like half yoga and half running. Similarly, if you decrease beta, you would get workouts that say, I'm just going to do bench press and bicep curls and nothing else because I'm a frat boy. And then if you increase beta, you would have more variety, and you might even be willing to say, I do weights, but this workout, this category does have a little bit of running, something like that. So the precise way of saying this is that those sparse priors that encode our qualitative assumptions are Dirichlet priors. And the reason we do that is because Dirichlet is the textbook example of a prior on a categorical distribution that can be sparse. We'll get to more reasons later, but the basic reason is that it's the first distribution that a lot of people think of when they want sparsity. So very briefly, It probably won't be enough to be self-contained, but it could be a refresher. Here's a crash course on the sparseness of a Dirichlet distribution. So here we're imagining that it's a three-dimensional, we have a prior RNA three-dimensional thing. So it has, this is component 1, this is component 2, this is the third component. We can draw it in two-dimensional space because all the probabilities have to add up to 1. So they're not, we only have 2 degrees of freedom. So what we do is we draw it on what's called the simplex, which is a triangle where one corner all the weight is in one category, in one corner all the weight's in the second category, and in one corner all the weight's in the third. And what you see is that as we adjust our parameter alpha, which is called the concentration, if it's big, you get the picture on the left where much of the weight of the Dirichlet distribution is in a balanced place where every one of the components has a significant probability. Then if you set it to 1, you get a uniform flat prior. And then in the case of LDA, what we do is we set it to a small value where most of the weight is on the corners or the edges of the distribution. So what that says is that if you take a random sample from the Dirichlet distribution, you're likely to end up somewhere like here, where almost all the weight is shifted toward one category, and then you have a little bit for the other 2. And you might get something like here, too, where 2 categories share some significant weight. But the point is that when this parameter is small, most categories you draw from will get hardly any weight at all. So let's take a step back and ask, can we answer the questions with this model? I'm not saying, do we answer it, because that would be you have to take a data set, run the model on it, and say you do or do not like the results. But in principle, does this model answer the questions we want? And so if we were trying to discover what are the different categories of exercise, then I would argue this model has the ability to do that because it has the sparsity assumptions we put in. And there's definitely enough of a signal in this data set that different types of exercises, different types of weight exercises occur together, different types of yoga poses occur together, that it should be able to learn those categories. We can also say that 2 people are similar in a way that we couldn't in the beginning. I mean, we could in the beginning if we can read, but if we can't read, if we don't have any prior information about exercises, we couldn't. But now we can, because let's consider the 2 runners. So yes, person 2 and person 5 don't use any of the same distances of running, but if we apply LDA to this data, we would realize that person 2 uses the category of running in much of his workout, and person 5 uses the category of running in much of her workout. So from the point of view of the raw data, they don't overlap. But from the point of view of the topic model, or the categories that we've discovered, they overlap quite a bit, because they're not both doing weights and they're not both doing yoga. So given this framework, we get a very large amount for free. We can solve a lot of problems with no modification whatsoever, because we have discovered a basic pattern of nouns are a mixture of a few nouns which have a few nouns. We have the 2 sparsity assumptions. That's why the word few appears twice. And we really just fill in the blanks. My kids would have a lot of fun with this because they would make potty jokes. But we're not going to do that this morning. We're going to fill in the blanks with more serious things. So the way we filled in the blanks so far is that workouts are a mixture of a few styles of exercise, which are composed of a few different types of exercises. When people think of LDA, they think of a famous paper by David Bly and Andrew Ng and Michael Jordan from 2003, which introduced LDA. Didn't really, we'll talk about that. But it introduced LDA to a broader audience, even though it didn't invent it. And what that model says is that documents, so just like a New York Times article or a paper in Science, is a mixture of different topics. A New York Times article might be some mixture of economics and politics and, I don't know, horticulture if it's the flower section. Does New York Times have a flower section? I don't know. And then every topic is a mixture of a few words that that topic really likes to use. So the economics topic would really like to use inflation and would really like to use unemployment. Now, where LDA was actually invented was in the context of population genetics in 2000 in a paper by Pritchard, Stevens, and Donnelly. And it's really almost the same model. The idea is that a human is a mixture of several ancestral populations, usually in fairly small amounts, just because there haven't been so many generations since humans stopped being geographically isolated. So maybe the Pritchard-Stevens-Donnelly model wouldn't work a few hundred years from now when people are just so mixed that sparsity is not accurate. But for now, it's still kind of accurate to say a lot of people might be like 90% Northern European and 10% Neanderthal. I realize that 10% is too big. And then every population has a few characteristic SNP frequencies. So then another example of filling in the blanks is in cancer. Because basically a tumor is made of several different subclones. Might also contain some normal cells that you biopsied by mistake. And every one of those has a characteristic set of mutations. So one subclone might have a different mutation from another one, and a third subclone might have another set of mutations. So if you sequence a tumor several different times, you can apply LDA totally directly and deconvolve it. Then a final example, which is what Sam's going to talk about in the main talk, is in the field of single-cell RNA sequencing. So the idea here is that cells are a document. They're doing some things. That's a very generous say. They have pathways. They're a mixture of pathways. And pathways is a really overused and horrible word. And I'm using it, and I'm a little embarrassed. But because I'm not a biologist, I don't know something better to say. So really, let's say cells are a mixture of things that cells do. So for example, a cell might say, OK, I am differentiating into biological process. Thanks, Sam. Programs, cellular programs. The reason you don't want to say pathway is, let's say you have a type of cell that differentiates into a neuron. And let's say that neurons, this is not going to be true, let's say neurons really need to ramp up their glucose metabolism, and they also need to ramp up their alcohol dehydrogenase activity. I'm making this up. I'm not a biologist. But the point is that those might be two different pathways, but they would be one cellular program, the program of things that a cell does when it differentiates into a neuron. So cells are a mixture of a few things that cells do, and every things that cells do category has a few specific genes that it might turn on or might increase the expression of. So it's the same idea. So I said that by filling in the blanks, you get a few more models for free, but you also, for free, answer another question or do another useful computational thing. So really, you've gotten dimensionality reduction. So again, consider the runners and compare them to the weightlifting person. So in the original space, let's say we represented a workout by our encoding of ones and zeros, or actually not ones and zeros, but if we have an exercise appear more than once, we'll give it a 2 or a 3, so counts. This is the bag of exercises representation. And for the sake of convenience, so I don't have to write a lot of zeros, let's say the first component is the 400-meter run, then the 800, then the 1600. So as we saw before, person 2 and person 5, even though they're both running, do not overlap at all. And so if we took the dot product of their vectors, we'd find they're totally orthogonal. If we took a distance in that original space, we would find that that distance is as long as the distance could be. And they are no more similar to each other than they are to the weightlifting person. Even worse, what if that weightlifting person did an 800-meter run to warm up? So then you would find that person 2, person 2, who does an 800-meter run, is more similar to the weightlifter than to person 5. So this is absurd. But when we do LDA and we get topics and we say that, OK, well, person 2 is a mixture that's extremely skewed towards the running topic, and person 5 is extremely skewed toward the running topic, then we would find that they are very similar. If we took an inner product between their topic vectors, they would not be orthogonal. And if we took a distance in the Euclidean space of topic vectors, they would be very nearby, much closer to each other than to person 1. So a very general and powerful thing you can say about LDA is that it takes you from a high-dimensional space, a useless high-dimensional space where the idea of taking a naive Euclidean distance is misleading, and it takes you to a low-dimensional topic space where Euclidean distance is not misleading. It's meaningful and it's useful, and you can do things with that. So I'd like to present a few examples right now of where LDA is used really just as a dimensionality reduction preprocessing step, but a very useful one, just to show you the power of this idea of moving into the space of topics. So one example, and I'm not stating that this is a state-of-the-art example by any means. It's a fairly old paper, and I imagine that convolutional nets would do a lot better for this problem. But pedagogically, it's useful. So the idea is you have a problem of protein-protein interactions. You're trying to classify, do two proteins have some affinity for each other that's greater than some threshold? And what the authors of this paper did is they first represented proteins as words. The way they did it is they said their words are 3 consecutive amino acid sequences. So if you have a leucine and an arginine and another leucine, that would be one type of word. Now, the number of 3 consecutive amino acids is 20 cubed, which is a little too big. So in order to simplify things, they didn't actually take amino acids. They divided amino acids into 7 categories based on their electrical charge and their dipole moments. And then they took 3 mers of those categories. So instead of 20 cubed different words in the vocabulary, they had 7 cubed words in the vocabulary. So they took that, and they said, they filled in the blanks. Proteins are a mixture of topics, and each topic has some characteristic frequencies of these amino acid 3 mers. So they threw that into LDA. They got topics. And every protein, therefore, gets a feature vector of which topics it's using in this space. So then they took that LDA preprocessing, and they trained a random forest classifier based on it. The way they did it is if you have 2 proteins, you take its LDA topic vector, you take the other protein's LDA topic vector, concatenate them, and then feed that into random forest. And surprisingly, they actually got a very effective classifier. I mean, I didn't read the paper well enough to judge if they were cooking the books somehow in their accuracy metric, but it looked surprisingly good. So here's an example that's more contemporary and more state-of-the-art in single-cell RNA. So one thing that people do in single-cell experiments is they take a huge number of cells. It could be thousands, tens of thousands, even hundreds of thousands of single cells. And they get the RNA expression data for each of these cells. Then one thing people want to do is find clusters of RNA cells, clusters of gene expression to get different cell types. But in addition to that, you might not want to just have the clusters. You might want to have the relationships between them in time. You might want to discover trajectories of cell differentiation or development. So what people did in this paper, it's called the cell tree algorithm, is they filled in the blanks, as we said before. A cell is a document. A topic is a cellular program or a cellular process. And the word counts in this case they're the log transformed transcript counts of RNA. Why they log transform is more of a specialist question. I don't have a good answer to that because I don't actually work with RNA. I'm just the messenger here. But they do, in fact, log transform it. And you could probably argue about whether this is a good idea or whether another transformation is a better idea. I won't get into that. They do some sort of transformation that, in their opinion, makes LDA work better. Then they apply LDA to all their single-cell data, and they get topic vectors for every one of their cells. And then, so as I said, when you go to the space of topic vectors, Euclidean distance becomes more meaningful. And so that doesn't just mean take the distance between two cells and answer, are they similar? That can mean doing anything that requires a distance metric. So in this case, what they did is they made a minimal spanning tree of all the cells in order to determine trajectories in cellular differentiation or cellular program space. If they'd done a minimal spanning tree in the original space of just raw gene expression counts for tens of thousands of cells, it almost certainly would have been meaningless. But in the small topic space, they get very meaningful results. And so what I've shown here are some of the spanning trees that they discovered for their cells, colored in two different ways just to show that it is meaningful. So over here, the way they've colored it is this is known truth data. They have labels for what these cells are. And as you can see, as they discovered this tree in their topic vector space, the colors change in a very continual way. There's a logic, there's a rainbow, basically, to how the colors evolve, which shows that the tree they discovered in LDA topic space is meaningful in terms of what cells do. Then the second picture shows the same thing colored by the LDA topics they discovered. And again, the basic point here is that it's a rainbow. It is a continuous evolution of color as the cells move through this space, which indicates both that LDA is useful for discovering something that has real meaning, and also the LDA topics are directly meaningful in and of themselves. Yes, Alex.
Yeah, this is awesome. So this slide brings a question that I had like 2 slides ago, in which you were saying Euclidean distance in the original space is not meaningful, but in the reduced space, Euclidean distance is meaningful. Now, I'm not a topologist.
I'm not either.
Right, but yeah, it's just a curious thing, like why is Euclidean distance or like the right or good distance?
It's not necessarily right. You could definitely think about using other distances. I think
you can
there was some paper I was thinking. There's some paper I read that was strongly suggesting some other distance on the topic weights. That of course I cannot remember now. Is it the right thing? And you can certainly use Euclidean distance in RNA space too. It's not like, I mean, people do it all the time. It's just,
I guess not the project. The only thing that comes to mind is, orthogonality, Euclidean distance, yeah, and so like sparsity, kind of like forcing the topics to be like near orthogonal. I don't know, but what if they're not that sparse? Like I don't know, something like that.
So I'll give 2 answers to this question, and they're not, neither of them are great, but they're better than nothing. So the first answer is that I actually lied, and this paper doesn't use Euclidean distance. It actually uses chi-squares distance, but the point is that it's related. What they do is instead of saying x1 minus y1 squared, they have, I think, x1 minus y1 squared over x1 plus y1. So they normalize the squared distances by the total sums. But I guess the other answer isn't so much that you get to a space where Euclidean distance suddenly becomes really good for this theoretically sound reason, as you get to a space where just any distance makes more sense because the original space was just totally stupid. I mean, really, you could say it's just a general property of dimensionality reduction, is that you lose the curse of dimensionality. As long as your dimensions have some sort of logic to them, then by reducing to that space, you've lost the curse of dimensionality. So especially for RNA, where so many of your counts are 0 due to stochasticity, but also dropout, Euclidean distance is just horrible. And if you consolidate things, at the very least, if you're doing nothing else, you're still just sharing the statistical strength for all the genes in one topic. You're hopefully doing better, but at the very least, you're adding some logic to it. So you could think of it as getting rid of the pathology of the high dimension if you don't want to think of it as doing something beautiful in the low dimension. Yeah, John.
So besides this chi-square metric you mentioned, and Euclidean distance, and maybe cosine, like the angle, are there other distances that people use that are particularly from the more information theory point of view? I mean, these are distributions. Have you seen people using, cost entropy, something probabilistic as a metric?
I have seen a bit of that. I'm not an expert on applications LDA. I'm not an expert on LDA either. I'm actually a guy who volunteered to give a primer talk because I thought it would be fun to learn about LDA. But in some of the papers that I've read in the last month or so, I have seen some more information difference. I saw one thing that looked a lot like Earth mover distance, but wasn't. I forget what it was. That's about all I can say, because that's the depth of my knowledge. There are lots of distances.
Hillinger, I think that's the symmetrized KL.
So just to catch our breaths right now, I would like to show an unrelated example that's just really cool. So if you've seen PCA talks and you've seen the PCA map of Europe, this is kind of the equivalent of that. It's just for fun. So there's a guy named Ben Schmidt, and he has a blog on digital humanities. And he was evaluating LDA. And so one thing he did was his specialty in life is 19th century whaling logs, because it's the humanities, and that's what you do. And no, but I mean, it's super cool. I'm glad he does it. And what he did was he took just the latitude and longitude coordinates in the whaling logs. So the whaling logs would be like January 7th, 1801, stormy seas, a great white whale tried to swallow the ship, latitude 48.79, longitude 42.93. And he just took those, and he just did LDA on the words of latitude and longitude, and he plotted how the topics cluster. And as you can see, the topics are pretty reasonable, surprisingly. Like, here's a topic where you go from North America, you round the Horn of Africa, and you end up in the Spice Islands. You have, seems reasonable. I mean, I don't know much about 19th century whaling, but this seems like a thing that a ship would do. And he claimed that more than just being lines in oceans, it was, in fact, more reasonable than that. He also said a few of them were totally absurd, but he said on the whole, it was pretty good. So, really cool picture. Now back to work. So you can extend LDA in a lot of ways. I mean, one of the great things about it is that it's very modular. You can just insert it in another graphical model to answer more questions. So I'll give you an example of some things that are done. It's not totally comprehensive, but the idea is just a bit of inspiration. So one thing you could do is that in a lot of these data sets, the way they evaluate LDAs, they might have a corpus of every New York Times article from 1900 to 2000 or something, and they feed it into LDA. But you could ask, well, topics evolve over time, don't they? I mean, if an article is about economics in 1980, it might talk a whole lot about stagflation. An economics article from 1910 is not going to talk about stagflation, nor is it going to talk about Keynesian economics or anything like that. So topics evolve. And that might be a really interesting question. So the way you do a dynamic topic model is you divide your data into time slices, like 1980 to 2000, 1950 to 1980. Every one of those time slices has its own copy of LDA, its own topics, its own probabilities for those topics. But you connect the topics. So from time to time, the topics are shared, but the word frequencies in those topics is allowed to evolve. And the specific way they connect them is with something that basically looks like a linear dynamical system. I'm not going to get into the details. The basic idea, though, is you share topics, and the topics evolve over time in some sort of sequential Bayesian model. One other thing you could say is that the Dirichlet prior is not adequate because it has no idea of correlations among topics. So for a Dirichlet prior, you're just as likely to say that this New York Times article is about horticulture and warfare as you are to say it's about politics and warfare. And maybe that's good enough. Maybe you have enough information in the rest of your data that you don't need the strength of that prior information. But if you do, you could replace the Dirichlet prior by a prior that has correlation. So the way this is done is you replace it with a logistic normal. And as you can see in these heat maps, it does some similar things to Dirichlet. It has the power to hang out near the corners of the simplex or hang out near the edges, but it also has correlations.
And the sparsity in the logistic normal, is it also criteria?
Yeah, you can make the logistic normal sparse. Because the way a logistic normal works is you take a Gaussian, which is not yet sparse, but then you take a softmax of a Gaussian. And a softmax it's basically like you multiply by a constant alpha, and then you take e to the alpha times component 1 plus e to the alpha times component 2. And the bigger alpha is, in that case, the more the exponential gets dominated by whatever component was bigger. So like if your Gaussian had a draw where one component was a little bit bigger, when you take the exponential inside the softmax, it'll drown out other components. So it has an adjustable sparsity, really for the same reason that a softmax can be made more or less sparse. Not exactly 0, no. Same as the Dirichlet. You're not getting any exact zeros in any of this. So there are too many papers to count about turning LDA into a supervised or a labeled model. So I'll just show the basic idea of it. So the basic idea is that a label for data is related not to the raw data, not to the words, but to the meaningful part, to the conceptually meaningful part of your data. And so basically, you can turn LDA into a supervised or semi-supervised or a labeled model just by putting an arrow between the topic vector and your labels, your known data. And the arrow could be going in either direction as long as there's some connection between observed reality and your topics, you get a supervised model. This is not a mistake. There is no picture here because the pictures for hierarchical LDA are terrifying, and they're really ugly. But I just wanted to mention that there is such a thing. So in LDA, you might have asked the question, what happens when we make our topics more refined? Like, what if we change the prior to say, I want 100 topics instead of 50? So maybe some of your topics would split. Like, for example, in our workouts, what if the running topic splits into middle distance and long distance? Or what if our body weight training with the pull-ups and the push-ups got combined with the weight training into a strength training category? So when you split categories, in LDA, because it's flat, you don't know they're related. So if you have two different running categories, you don't know that they're two running categories. They become completely distinct, and you lose that information. So the way to fix that is instead of having a flat prior or a flat structure of categories, have a hierarchy of them, have some sort of tree. And it looks really intimidating and complicated, and it is complicated, but you can actually do it. So I'll now issue a few warnings and caveats about LDA. I want to be clear that LDA is probably the best topic model out there. It's really good. If you're trying to do a topic modeling, you should always at least think about it, because it's very effective, but it's not perfect. So here are a few things that make it not perfect. So first, common words are really, really confusing to LDA. So if you're considering topic modeling with New York Times articles, let's think we've been talking about stagflation, and we've been talking about complicated words. Let's talk about simple words like prepositions, conjunctions. If those are left in your data set, your topic model is going to get really confused, because it will say something like, oh, hey, whenever I'm talking about economics, I see the word in and of a whole lot. And whenever I'm talking about horticulture, I also see those words in and of. Maybe those topics are related, so it gets confused. The best case scenario is that your signal just gets diluted and you learn a slightly worse model. The worst case scenario is that your model becomes totally stupid. So by far the most common pre-processing step in LDA is to remove common words. Or in the case of single-cell data, you would do something like remove housekeeping genes from your data set.
I'm going to protest.
Oh, good.
But I can protest in my own thing. I think it's really good to think carefully about all of that. And I don't know that it's the best analogy, partially because we don't really know that much on the cell end about exactly how stable those things are and how much signal they co-occur with. And the second thing is that I can imagine that it's inefficient, but I'm still, after like a long conversation with Alex and Dylan and stuff, I'm still mystified as to why it doesn't just pull out a big topic that has a lot of trash in it and then go find other ones. Like, to me, it's just not clear that it...
It's like the trash query makes process.
If it correlates with more specific things, then it's not trash.
Well, but then the specific things, even though they're separate from each other.
Why would that be the case? Like you can just make additive, it's additive, right? So you can have other topics that, I don't know. For me, it's not clear at all why this fails, and I feel like I have read about it, I'm still unclear about why this, I can imagine it's inefficient, like that you don't want that, and you could get rid of it, but you can, I don't know. Anyway, I read this thing that was talking about maybe you could get rid of that, some of the words afterwards.
It's not
So
as
you
clear as
could
I'm making
potentially
out
also
to be.
look afterwards and see what the signal looks like, at least for biological things where we don't know that much about it. We know we don't care about in and dot. You know, we know those aren't meaningful. But like, for example, if you were comparing American, I don't know, if you're comparing different language, for example, you might not want to throw out those kind of words because you might want to know if, you know, they don't have articles in some other language or something like that, right? Like there are cases where the common words aren't totally worthless.
I do agree with you.
Yeah.
And I would say common words aren't a guarantee that bad things will happen. It's just, it becomes less predictable and less interpretable. Because it's really easy to say that this topic really likes these words, but it's...
I'm so sorry.
Oh,
Okay, will you just... Sorry.
I'm gonna suggest we do it for discussion, or
Okay.
you can do it part of the
No, but I just, nobody could hear what I said, so I don't know if he wants to...
Yeah, I think I should at least... So the question was, does it necessarily do bad things to keep common words? And Sam was arguing that it doesn't always, and I agree. Just long story short, is I agree with that. It doesn't guarantee horrible things. It's just a risk. And it's a risk that people don't understand in a very great theoretically founded way. So it could happen. And there's no shame in keeping common words, keeping housekeeping genes, and then qualitatively saying, I don't like my results, I'll throw them away, or qualitatively saying, you know, I'm happy with these. Another thing to keep in mind is we know exactly what LDA is doing. It's not like deep learning. There's no black box here. The only signal it looks at is co-occurrence of words. Or in single cells, all it's doing is looking at genes that are turned on at the same time. That's the whole signal. So it's not more magical than that, and it's also not doing less than that. Finally, the dirty secret of LDA is that usually at least some of your topics are really, really garbage. So in most applications, you might say, I have 100 topics, and you can probably expect the first 20 to be good. You can probably even expect the first 50 to be good. At the bottom of the barrel, it could be totally meaningless. And I don't have a good reason for this, but I have a hand-waving reason that might be good, which is let's imagine you were doing LDA in a greedy way, however that would be. So you made the first topic, and you made it really good. It's super coherent. And you made the second topic with sort of all the words that don't seem to be explained by the first one. And you go on. And as you're running out of topics, what you're doing is kind of making topics that can't be totally coherent in and of themselves because they also have to explain the data in your model. So they also have to explain all the words that aren't explained by the other topics. And that competition between having to fit your data and having to make a topic that makes sense clash. And eventually you will inevitably have some topics that are fitting your data at the expense of making sense. Now, there's a lot of QC measures, a lot of ways to decide whether your topics are good besides just inspection. People like Hannah Wallach and David Mimno have done a lot of good work on that. So it's not like you're sunk. You can query your model and say, I don't believe in these topics, and throw them away. I don't think I'll do this slide. I think if people want to ask those questions, I could answer these questions. But I didn't want to end on the warnings page, so on here, because LDA really is a genuinely great model.
Okay, hi. Just want to get a quick read on who's here. Does anyone know any immunology in here? Awesome. Okay, that's what I was thinking. That's perfect. So first, I just want to say right away, so I don't forget, that this is a collaboration with an experimental lab with the Flavelle lab at Yale. So I don't do experiments. None of these were done by me, but it was great. Okay, so I'm going to start off by showing data right away. It's very awkward. Like I can't see my, all right, I'll look here. This is my data here. This is data from a paper on retinal cells by somebody in our lab from a couple years ago. And so these are innate lymphoid cells. And you can imagine that this could just be like a bunch of noise. And after struggling with it quite a bit and trying to vet in every possible way, I decided that this is not just totally noise. There is actually interesting structure here. There's interesting immunology that you can learn. And I will properly introduce this, explain the labels and everything else. But I just kind of wanted to put out there that, like, initially, single-cell RNA sequencing has been about categorizing cells. And a lot of the times, that process looks like this. You get these really nice, distinct, kind of homogeneous clusters of cells when you look at the data. And you can imagine that the techniques for analyzing this kind of data are very different from the techniques that you might want to use for this kind of data. Although next week, Dylan is going to talk about some cases where things look like this but are actually more complicated. And so it's this kind of LDA type approach can also be useful even when you have this kind. But today we're going to talk about this continuous states. So let me do a little more formal introduction. Okay, so usually I say, okay, I work on innate lymphoid cells, and I just tell you that they're like T cells, and then that helps everyone. But I'm going to assume that that's not as helpful as it could be here. So I'm just going to give you a tiny bit of background because it's useful for understanding why people have certain expectations going in about these cells. These innate lymphoid cells they were not known at all a bit more than 10 years ago, so they're relatively recently discovered, and they basically were put into this framework that we already had for T helper cells. So T helper cell, they have these naive T cells that travel around, and they have these antigen receptors, and so then they can interact with an antigen-presenting cell, like a dendritic cell, and then they see a specific peptide, a specific piece of foreign antigen, and they respond to that. They get activated, and along with some additional signals, they go in one of, okay, now this is 3, it used to be 2, this is probably growing the number of directions, but they go down a kind of specific differentiation pathway. They have, these pathways are marked by specific transcription factors, they express characteristic cytokines, and they do a specific job this way. They fight an extracellular pathogen or worm or something like that. So this was kind of the backdrop of the scene when so here, this type 1 is good for fighting bacteria and viruses, helminths, extracellular bacteria, et cetera. And we learn more about, this is a very high-level summary. There's more nuance here, of course. So then innate lymphoid cells came into the picture. They're called innate exactly because they don't have a T cell receptor. So we don't think of them as responding to a very specific antigen. It's a little bit misleading because they do actually respond differently to different kinds of pathogens. So this is kind of the ambiguity in the naming. But they sit there in the tissue. So they are resident as opposed to circulating. They don't have the receptor. They're still, what people realized when they first kind of got discovered is, hey, look, like these seem to be divided into these subsets where they express specific transcription factors and specific groups of cytokines that we associate with certain types of immunity. They contribute to those same kind of immune functions the same way that T cells do, but because they don't have to go through this process of responding to an antigen and multiplying and stuff, they're just really fast. So maybe they're like the EMTs that are sitting there, I think I even say that, yeah, they're like the EMTs that are sitting there ready to respond, ready to deal with a threat, and kind of manage until T cells that are more powerful come and take over in a couple of days. And that's still one of the kind of basic models for them. Increasingly, though, we realize that because they're sitting there, they actually can do a lot of other things. They can help regulate homeostasis. Even when there's not a threat, we think like this function is broader. But that's kind of the backdrop for like what I'm going to show you. Okay, so, oh yes. And then, of course, because they do important good things, when those things don't work right, when they get out of balance, they also contribute to disease. So we know that these different subtypes, just like T cells, are related to different kinds of pathologies or pathological inflammation. And the one that I'm going to focus on today is in skin. This is a psoriasis model. And psoriasis is basically when you get, the short version is when you get really bad thickening of the skin. So your skin just starts kind of going out of control. So let's look at a model for that. This is my extremely oversimplified picture of what we know about psoriasis. So we know there's some trigger that we don't particularly understand. We get signaling that I'm not going to talk about to this dendritic cell, and then that dendritic cell signals using IL-23. And originally, we basically focused on T cells, and they start producing these cytokines, which I'm going to name because I'll probably bring them up later, forgetting that they aren't meaningful. So IL-22 and IL-17, these are specific cytokines. They're called type 3 cytokines because they're produced by T cells and ILCs in that third category. And they cause keratinocytes in the epithelial tissue to just start proliferating. And then those cells also send back signals that make this process kind of spiral out of control. And then we realize, oh, okay, ILCs are also capable of producing these cytokines. They're also sitting there in the tissue. And so we can see them producing things like IL-17 and IL-22. They also seem to be contributing to this process. And on top of that, they can communicate with T cells. We know that they do things like amplify the T cell response sometimes. So maybe that's their role. Now, the only question is, like, at steady state, we only see ILC2s. This is what was kind of known, that when you look in at the skin, just sampling what ILCs are there, it looks mostly like they have this ILC2 profile. So then the questions were, where are ILC3s coming from? Do they get recruited in, which has been shown in some contexts? Do they get expanded from some tiny population that we haven't seen yet? Do they actually come from some precursor that's there? Or do ILC2s become ILC3s, which I realize to this audience sounds like a perfectly reasonable possibility, but to immunologists is like, blows their mind. So, I mean, at least for some of them. Some of them are more used to thinking about that. So it's certainly paradigm breaking, say. So, and all of these are scenarios that have already been shown to contribute to pathology. So we know that these things come up in different contexts, different tissues in different contexts, so it was not clear. So this is the model that we used. Obviously, this is the part where my experimental collaborator was dealing with these details. But we have a mouse model. We used, I'll show you a little bit as we go through. We used wild-type mice. We also used mice that were genotyped to have certain deficiencies. So some of them don't have any T cells. Some of them don't have any gamma delta T cells. Some of them don't have either T cells or ILCs, just so you can kind of try to piece apart the contributions of these different cells. Then you give a injection into the skin of the ear of IL-23, which is that signal that they get from dendritic cells. You do that every day for 5 days, and every day you're measuring the thickness of the ear. It's growing and growing, and then you take a sample, look at it histologically, isolate the cells from the sample, and then we do a bunch of downstream analysis, including one of these paths is that you sort them, you isolate the ILCs, and you send them for single-cell RNA sequencing. Okay, so most of my part is on that. So just to convince you that this, you probably don't even care, but it feels weird to not convince you that ILCs are actually relevant. They're relevant. Basically, this is what you get if you have only ILCs and no T cells. This is the amount of thickness of the ear. If you have everything, this is what it looks like. And if you just like if you take a mouse that doesn't have either and you add ILCs, you get like from there to there. So basically, they're contributing a good amount of the pathology, especially early on. And that made it like kind of relevant to study this process. This is basically just showing that if you try to block a receptor that is known for homing, for causing circulatory cells to come to be able to exit their lymphoid vessels and move around, then you don't get any change in the phenotype. And what that suggests is that these are not circulating cells. So there's actually the cells that are sitting there in the tissue. It doesn't rule out all possibilities, but this was an experiment that had a big effect, say, in other kinds of inflammation. In the lung, it makes some of these cells, some of the inflammation just disappears if you do this. And that wasn't the case for us. Okay, so now we can go to data that probably interests you more. So here I'm showing the results from the single-cell RNA-seq in a TSNE. So every dot here is a cell, and here I have untreated and induced, and here I've got no T cells and T cells. And you can see that we do get a few specific kind of clusters, right? We have, let's see, we have this cluster right there, and it turns out those right away you can see with like very little brain power that those look ILC3 like they're really strongly expressing IL-22, they're probably driving the pathology. Then you also see that you have this, I'm just putting this out actually as a preview for Dylan's talk. So you get this cluster here, which have cycling cells in it, and people who analyze this kind of data know that you often get all the cycling cells off in a cluster. It doesn't mean that cycling is the only thing they're doing or that they don't belong to other clusters. And Dylan's going to talk about like how to piece that apart. I'm going to focus on what we initially called the blob and then renamed the cloud because it sounds nicer. So here, this is 81% of the cells. And like I said, you could imagine that this is a lot of noise and things like that, but I can say I tried most of the tricks that anyone I knew had tried, and I didn't feel like it was only that. There's definitely some noisy processes, though, yes.
Sorry for my ignorance.
No.
What TC you want to do?
Oh, absolutely. Thank you for asking. I just never want to bore people, but I should definitely explain. So this is like the T-stochastic neighborhood embedding. This is the dimension 1 and dimension 2. It's just a nonlinear dimensionality reduction technique. So it has the property that cells that are close together here tend to be close together in high dimensional gene expression space, but it's not linearly scaling. So if you see two things far away from each other, that doesn't necessarily mean they're really far away from each other. It's a way of trying to capture local structure in 2D. So it has to kind of bend the space. And they're really, really commonly used in single cell because people like to look at their data. And it's in very high, we get 20,000 genes or something out. And so it's common to kind of do some kind of even PCA first and then TSNE to be able to look at it. What this means to me if I see this is that the density of cells in some high dimensional gene expression region is fairly even. So that could be like there's a process that's like kind of spread out over a bunch of different gene expression, kind of a region of gene expression, and it's like fairly spread. If you had a really strong density in some place, that would come out like a cluster in TSNE. It would break it up more, and that could just be from noise. Yes.
But
yeah.
There are very simple algorithms to find clusters in high-dimensional spaces.
Yes.
You cannot visualize them.
Yeah.
Why to enforce the analysis to 2D?
We don't. I never do any analysis. I just show it in the TSNE because people like to see it there. So I would never run clustering in TSNE. The clustering, anything that I do is in high-dimensional space. It's just people tend to look at the results in the 2D. I totally agree with you.
But would you do the clustering in 20,000 dimensional space?
No, usually it's some reduced dimensional thing that makes it, there's also a lot of noise, like everyone has said. There's a lot of noise, a lot of zeros, it's sparse, so usually it's some combination. This is like a whole other topic in and of itself that we could spend a while on, so let me just keep moving, but I'm happy to talk afterwards about that. So, of course, you can cluster anything you want to cluster. So, we clustered this. And part of doing that meant that we were able to kind of see, first of all, what characterized these clusters. This is just using a k-nearest neighbor graph kind of clustering algorithm. You can do other ways. It's like this structure is basically you're just ending up discretizing continuous structure. And you can see that when you look at some of these kind of key genes, like this is a type 2 cytokine, and we see that it's like kind of here. It's also in this ILC3 cluster. Then we see this, sorry, am I totally blocking your view? So we see KLF4 is a transcription factor. It's repressive. It seems like it's kind of emphasized in one area of the region, but it's kind of got a continuous gradient. AREG is another type 2 cytokine that's it's like important for healing wounds. And we expected this to be really like kind of specific, and it wasn't. It was kind of everywhere. Anyway, you do see, of course, some specific expression. Like I said, I'm going to ignore this cluster here as I already have been because it's only specific to the RAG. And I don't particularly care that much what happens if there's no T cells because that's not like physiologically relevant. Although it could be interesting. So basically, we wanted to use some kind of method to try to better represent this kind of continuous state. Let me go back for 1 second. Like if the best we can do, this is kind of like pixelating, which isn't necessarily bad, right? You can cluster, you can characterize the clusters, but in the end, the thing that you're representing is some kind of continuous process, continuous state, and it made sense to me to try to represent that in a way that was interpretable. Maybe there are different functions that are more important to cells in different regions of this, but they're kind of a mixture of these different functions. Does that make sense? Have I lost anyone? Okay, great. So So the way that we did this was using topic modeling. And yes, yeah.
I really enjoyed learning a lot of biology just now.
Okay.
So in the same way that we get upset when people apologize for math in
Yes.
the other seminars.
Don't apologize for
Don't
biology.
apologize for biology in the
Okay.
seminar, because
Okay.
it's been really helpful to orient why we should care and also to understand what some of the specific challenges
Right.
might be in
Right.
this kind of data.
Cool. I think actually that one of the things that I have observed from doing single cell analysis for a while is that people have very different kind of data challenges and analysis challenges depending on the context they're working in. And so there's not really like, I mean, there are a few central problems, but in general, you know, what people get hung up on, what they're frustrated about in their data, what they don't feel like they have the tools to do is very specific to the setting that they're doing it in. So even in immunology, you know, I'm obsessed with this continuous state thing because I work on ILCs. If you work on some other process, it could look really different, and you might not care. Although I think this is going to be increasingly relevant in immunology. There's a lot of evidence that what people have done in the past, right, is they used surface markers to sort cells. So all of the cells are kind of categorized according to things that make them easy to measure and find, as well as other kinds of genes. And I think that that has also kind of influenced what we know about them, right? And so now that we can look at them this way, we're getting a better view of like this underlying more continuous space that it was hard to sample before. That's my 2 cents that you can ignore. Okay, so topic modeling, I guess there's a chunk of people who are not here for David's primer, right? Yes, okay, great. So I will just review it. I don't want to bore everyone, but I think it's good to think about the background and in what ways it's analogous or not analogous. So it was developed, like David said, it was developed actually in the context of admixture estimation, but I'm going to talk about it here because this is how it's like very well known. It's used in document analysis. So say you have a collection of electronic texts and you want to characterize them in some way that is meaningful for you. You want to understand kind of what they're about. And so you know that text can have multiple different topics, and those topics influence directly like what words appear in the text. So you can imagine that you can basically infer by looking at the frequency of different words that occur in the text, you can kind of infer what the topics that pervade a whole collection of texts are. So here this is just like they basically went through, here we have these topics. Topics then, instead of being something abstract, they just become a distribution over words. So, this is gene topic, and this is life topic, and this is brain topic. That's how I name them, but really they're just a distribution over words that are related to those things. And then here we have each word in this document is then being assigned as coming from a specific topic. And you can have a mix of topics present in a text. So the goal is basically you have these documents and you want to infer what the topics and what the weights are, how much of a mixture is a document really about one topic? Is it about a few different topics? What are those topics? You want to do that for the whole collection. And then that gives you a representation that is really condensed and that you can work with. That's also interpretable by humans to some degree at least. Okay, so on a more technical way, I think it's really useful. One of the reasons I really like this is because there's a generative model behind it, and that has 2 advantages in my mind. One is that you can think about the process that generated your data and whether it makes sense, and you can alter that. So for example, this has been extended in a lot of different ways. I'll talk about a few of them briefly, but as we get into other scenarios, you can kind of tweak the different distributions that are here, which is harder to do if you just have an optimization kind of representation of the problem. So really, it gives you a matrix factorization. That's what you get in the end. But the way that we think about the generative model is that we have some number of topics. That's the trickiest part. You give it as a parameter. Then you have a Dirichlet distribution that is, so you have a Dirichlet prior over the topic distributions. If you don't know what that is, or that feels like scary, it's really just thinking about like what would make sense. You want some kind of distribution over discrete a discrete assignment. I'm going to assign each cell I wanted to assign each cell a probability that a topic appears in that cell. now I switched order cells, each document, whatever. And you want to do so in a way that's kind of sparse. So you don't want to have a topic be assigned a whole lot of, a document be assigned a whole lot of different topics. Because typically, unless it's like the whole encyclopedia or something, that's not going to be relevant, right? So here so you basically imagine that your topics already exist. And so, I mean, okay, so which way is this characterized? Sorry, draw a distribution of words. Okay, so each topic is then created by drawing a distribution over the words. So you first draw the parameter for the distribution, also from a Dirichlet, and then eventually you're going to model the actual topic assignments of the words as a multinomial. And then for each document, you draw a vector of the topic proportions, like I said, from the Dirichlet, and then you, for each word, basically figure out, you assign that word to having come from a specific topic. Okay, so in the world of single cell, I'll come back to this, but basically you're assigning each gene as coming from a specific process or function. And it's not even each gene, it's each gene count, right? So a gene could be present for multiple reasons, it could be performing multiple functions, and that's really important because we often have that situation. Okay, so why would we do this? In general, non-negative matrix factorization makes a lot of sense when you have count data, positive data, because it's more interpretable. It makes more sense that you can add two processes and get your result. Otherwise, it's kind of hard to interpret in PCA, say, what does it mean to have negative gene weights? We try to interpret those things, but I think as a model, as a generative model, it makes more sense to imagine positive components and positive weights. So the most important things are about the fact that you can represent both mixed discrete states and continuously varying states. Those end up with the same representation, but in your data, they look very different, right? So in your data, you might have a cell that belongs to two different kinds, it has two different identities that you want to capture, or it could be that you have cells that kind of fit this whole range of identities, and this is a useful model for both of those kind of scenarios. So basically, LDA puts this Dirichlet kind of prior structure so that you have a full generative model for the whole process, but basically, in the end, you're getting some kind of matrix factorization. And some of for me, one of the things that I'm really interested in going in the future is these kind of other extensions. Yeah.
So in the case of documents, a word is a word. It's either there or not. But in the case of cells, genes can be expressed at different levels. Do these sort of binarize it, like
no.
expressing?
No, because words are also expressed at different levels. Words appear more times than not, right? So what you can imagine is, actually, the way I think of it is like unfolding the gene expression matrix, and so that you just have the genes, like if you have a count of 10 for AREG, then AREG, it's like it appears 10 times in the document, kind of, right? Does that make sense? And because the thing is with LDA is it doesn't care, the vanilla version doesn't care about word order. It's very different from like kind of embeddings where they take into account context of words, right? So it's just considering you could order basically all of the, times you see brain that can all occur in the beginning, right? So it's just like a gene expression matrix in that sense where you've just ordered it. So I can come back into this, or we can talk about it afterwards. Yes?
Sam, I don't know if you wanted to make a comment about this, but I thought that's what Mertash was going to ask.
Oh, okay.
When a document is a bag of words, we can count all the words that the document is made out of. Probably there's not going to be trouble.
No, I think actually, in terms of the multinomial, right? So the distribution that you get at the end is a multinomial, which actually represents really well what happens when you have a cell, right? It's just a sampling of whatever the expression is that's there. And you don't have to deal specifically with zeros. You don't care whether it was sampled or whether it's really zero. It's just a representation of the sample that you actually got. So in that sense, you're not constraining, say, like with Poisson factorization or something where you're constraining the marginal to actually fit a Poisson, which we know doesn't happen. Like here, it's like, I think the multinomial really takes care of the fact that you just sample from what's there. You could imagine even a document as being like, you could model the first 10 pages of a Proust novel, and that would still give you something meaningful. It doesn't have to be like, you could imagine taking a sample of a document and modeling it, and that would still be totally valid representation of it.
The representation of the sample document.
Of the sample document, yeah. So that's how I think about it. I mean, I think it's like the data generation process completely matches what we actually do. So to me, it's like a rare, fair model. Okay, so, all right. So this is a schematic of what does it look like when you apply it to single-cell RNA-seq. So you get a bunch of, so each topic is represented as a distribution over weights. The way that I'm gonna, here I'm kind of showing this schematically, like you have the highest weight genes on top and going down. Yes, please.
Just beating up on how well it matches, and you're saying it's a perfect match to it.
I don't say perfect match.
Well,
I
want to press the boundaries.
Yeah.
Does this allow the appearance of topics and other topics appearing?
No. So so this is, wait, hold on, see? Correlated. So I think one of this is an extension that's been done. The reason it isn't used that much, it makes total sense, right? That you don't expect, like the examples they always give are like, if you open a novel, you don't expect it to be both about, neurons and romance or something, not necessarily. Anyway, so like could be, it's less common. Certain topics are gonna be more likely to appear together, which is absolutely true in biology, absolutely true in literature. So there is a lot of work actually on relating. So one that I think would be really useful for single-cell RNA-seq is a kind of a hierarchical model where you're allowed to have still topics that kind of come from different parts of the tree, but there's been a nice development where they kind of constrain the way topics are chosen to being passed on a tree. And so then you get kind of a hierarchical relationship, which I would like to just try and see. Then there's just plain correlated topics where you're allowed to have dependencies between how likely a topic is, if it's, if there's another one present, which makes tons of sense. They use I think it's like a log, is it a log normal? They use some kind of different prior, you can't use a Dirichlet prior, and then that makes the inference slower because you don't have the conjugacy. So basically it's just, I think it's mainly computational. The reason not to do that is that it gets a little harder to infer the model.
But what's different about cells as compared to novels or documents is that cells come from other cells and pathways, so this hierarchy and relation of topics and context dependent might be learnable from single cell data as well.
I mean, that's not different from documents particularly. Maybe it's even more, I see what you're saying.
Well, I mean, documents...
I think the topic structure can be very hierarchical in documents too.
But I think there's actually...
But maybe not the generation process.
The generation process. Documents aren't generated by differentiation from other documents. Right.
I think the question is whether so because you get kind of this additivity, it's true that you will sometimes, it's true that it loses a little bit in terms of being able to infer dependencies between topics, but I do think, and you'll get, I have to try it out. I just want to try it out to understand whether, did I just say something?
It doesn't yet include it, and it might, that's cool.
Yeah, no, I think these models exist, and they've been tried. They're just a little bit more slow or a little bit less accurate or whatever, totally worth trying. What I did was like try a thing, it worked, and then nobody would let me do anything else because I got something that worked, right. So now I need to try stuff out, other stuff.
It also just may be the case that as we have more data, a model with greater capacity to include these correlations or find hierarchies becomes useful as opposed to just overfitting the smaller data. But the challenge, like you mentioned, is that once you go to log normal, you lose conjugacy, you have to use play gamma algorithm, you have to use fancier inference algorithms to deal with scaling that computation.
Exactly, So I think, but we used to think that you couldn't do anything Bayesian on this kind of data because it would take forever and, then variational inference and other kind. Like, you can always hope they'll think of something smart to do. Sorry, did I just give you a heart attack?
But it's also not there might be an infinite number of documents you can use because people keep writing more documents, but we're gonna have a finite number of cells and cell types that we can use. And so we may have reasons to want to...
That is ridiculous.
Why?
Because there's going to be, every time you have a process, right? Like any kind of inflammatory response, any kind of stimulus, the context, like if you really...
I don't buy it.
Oh, come on. It's just like every book has been written already.
That's called a large number, and it's going to max out at some point. And I'm worried that...
I agree with you in principle that the major directions are going to be found, but I think, so I guess it's from studying, I think when you think about it anatomically, that's absolutely true. When you think about response to the environment, That's where I think it falls short.
Sure, but it's the anatomical where I want
Right,
to
but I care about the response, right?
Oh, that's
I care about.
But either way,
Yes.
the hope is
Carry on.
in finite amount of time for evolution and modularity in biology, that even the continuous phenomenon can be understood in terms of mixtures of discrete things, operating different levels.
Absolutely.
So it all, in the end, can be understood in a computer.
I'm just saying that
There could be very much
structure-
finite directions that are still useful for understanding area response. Yeah, we're just gonna.
You well answered the
Yes.
question.
Okay. I didn't even ask if everyone heard Aaron because I just assume everyone heard it. Okay. So where are we here? Okay, so I just wanted to point out that, I mean, this is intuitive probably for a lot of you, but here I'm representing cells as these are kind of the transcriptional profiles of cells, and I just organized them by, like, you know, this one has a dominant component from topic 2, this one and a little bit of topic 1. This is just like according to which one is kind of dominant. And here we see one that has a little bit of these and more of this. So you get, this is kind of what we hope to get out of it, is some kind of mixture profile for each of the cells, and then some kind of topics where we see like really nice familiar immune genes that are kind of interpretable as processes, as immunological response processes. Okay, so, and then basically by knowing about our genes or maybe doing validation experiments or testing things, you can start to try to characterize what these topics actually mean. Like you can give them a name, you can think of it, but it's obviously, you know, we don't start knowing these, right? These are completely inferred from the data, which I may not need to say to you, but that's not intuitive for a lot of people because many of the kinds of factorizations that people use now involve knowing your components ahead of time, knowing what like gene modules are present. Okay, so this is just me having tried a topic model with 15 topics. I want to say immediately a few caveats, okay? So first, I tried different numbers of topics, and I will go in 1 minute about how I chose 15 specifically. Second, this is not a demonstration of exactly how you should do LDA in your setting, because really I was just like, I think I'll try this out, see what happens. It seems kind of useful. And then, you know, I didn't know what I was doing. I had never tried it. I didn't know anything about, for example, removing common genes or things that I know now about. And probably if I went and did this, I would do this a little bit differently this time. But it's just a demo of the principle of why it's useful. The other thing is that Dylan will talk a lot about the fact that there's randomness involved. And so you should probably not put a whole lot in store with like whatever you get out of it. You could try multiple runs of it. You know, you can look for consistency. I did none of those things. Okay, so, but what I would say is that, sorry, just one more second. I would say that in general, I mean, even having talked with David Bly, who's the one who made LDA, I think the way that I think about this as being useful is for generating hypotheses and for exploratory representation. I think it's fine with me if a lot of these are not particularly useful or interesting. That's kind of true about like 90% of what I do anyway. Like what I care about is that I can capture some of what's interesting, some biology that helps me understand what's going on. I can validate, I can make my experimental collaborators do experiments to try to like help me understand if it's real. Yes?
Why are you still using TSNE in the gene space and not in the topic space?
Because, so first of all, this is, okay, so this is a complicated question. Why did I? Yes, so his question was, why am I still doing a TSNE in the gene space instead of in the topic space? Okay, so first of all, here, I'm showing the topic weights, and I'm still using the same TSNE because that TSNE is something that we understood something about at that point, right? And so then people want to see how do the weights relate to that TSNE that we've understood a little bit. Does it capture regions that we kind of had already tried to characterize? Does it look the same or different from clustering? So this is kind of the initial way of testing LDAs, comparing it with some explorations we already did. So that's part of why it looks like this. Second of all is that, you know, I actually did do TSNEs using the topic weight vectors and things like that. So I did do that. And I didn't find it like, I wish I had included some of them. I didn't. They didn't like radically change the picture. They defined it a little bit more, but I didn't get I mean, the point is a blob is a blob. Like there's still, you know, you still have a lot of mixtures. And so it like is a blob with more pointy parts. It wasn't like.
TSNE is just a way to visualize prior information.
Yes.
What did you mean that the TSNE wasn't informative?
Did I say it wasn't informative?
Yeah, you said that it didn't work, or I can't quote you.
No, it wasn't more informative. The TSNE on the topic representation was not that much more informative than TSNE on the original
it didn't, using the topic weights as input to the TSNE didn't like radically change the picture of the TSNE. I changed the little parts of it, but it wasn't, it had slightly different features, but it wasn't.
But if the cluster exists, it's a problem with Disney, not with the clusters.
The clusters don't exist.
The clusters don't exist. That's the issue.
Yes.
But topics are supposed to be like a cluster. You cluster.
No, so so this is a great point. Let me go a little further and then we come back, okay? Like I'll try to leave a couple of minutes. So let me just go through the kinds of topics that come out. I think that will be helpful right now. So we do get, like you're saying, we do get some topics that are cluster specific, and I show this partially because people want to know that they're not going to have to then go do cluster-based differential expression analysis. This basically subsumes that. Like I got clusters that were very specific to each of these, topics that were very specific to each of these clusters. I got like my cycling cluster that had all my cycling genes. I got my ILC3-like topic. I say like, and I'll come back to that. It's actually a little bit more mixed than this, and then I got this RAG-specific one that looks ILC1-ish. And then, and all of so this is just a density computed from the weights. So this is topic 7 weights across all cells. So the marginal across the cells, and these look very spiky, right? They're 0 almost everywhere except where you have a cluster. So that's one kind of topic I get out. Another kind is this sort of regional enrichment, right? And this is part of what I think is interesting, because instead of forcing me to, say, cluster and divide arbitrarily my signal in my cloud, I can instead characterize it with these kind of functions that are enriched in certain parts of the cloud, and they have like continuous weights that show me which of the cells are more enriched, and they can be overlapping in different ways. They don't have to respect cluster boundaries, and I'll compare them to clusters in just a second, but I think that's actually like a very important part of why I did this, was to try to find and characterize and explore the structure in this cloud, okay? So, I can come back to these in 1 minute. The last two slides are just about structure, or just about the topics that kind of weren't that great for my purposes downstream, but I think they're still real, at least in some degree. So one of them is this topic that was completely driven by ribosomal genes. Okay, so for most people, this is the kind of stuff where they say this is just trash, I'm gonna throw it out. It's technical quality, right? Because if you have cells that you sample less well, they look smaller, or if you have cells that are unhappy, maybe like this one, for example, was completely driven by mitochondrial genes, which cells start expressing if they're not like very healthy, happy cells. So to some degree, this is definitely a little bit related to cell quality. And I think it's also actually a useful view, because what we tend to do right now for kind of, QCing our data is we pick some threshold for, say, the number of genes or the number of UMIs or the percentage of cells, the percentage of genes that are mitochondrial, and then we take all the cells that meet that threshold and we call it QC'd. That's what I had already done. And I think it's kind of nice to see, oh, this is actually like a real distribution, and I could even use that distribution, right, in my analysis. Or at least I know how strong it is and where it is. I know something about that component as opposed to just like thresholding it. Anyway, it's not a big shock though. And then the other thing is that like this distribution was skewed very high. Like all cells basically had some component of this, and that's exactly the common word thing, right? Like they all express these ribosomal genes. They just have different amounts of it. And what's kind of unintuitive to people who don't work with this data is just that small cells have more of it because they have producing less other stuff. So immune cells that are not activated have actually a higher percentage of ribosomal cells. And so along with being kind of a technical thing, this is actually a signal in my real data. This is a real biological signal for me, and it always is in any kind of immune activation response, because the cells, when they get activated, they start making lots of cool immune genes, and the percentage of ribosomal genes goes down. So for me, those two signals are almost always correlated, and a signal I'm interested in and one that is kind of technical. Sometimes they're slightly different, but that's part of why I actually want to represent it and I care about it. I don't always consider it trash. Then we had some that I just can't interpret. My friend doesn't know. They look very diffuse, so I didn't worry about them. Like they're kind of ribosomal, kind of other stuff. Didn't know what they were. There are a couple here that I also didn't use but not because they're necessarily trash, just because I really couldn't interpret them. So these were dominated by a single gene. Now that can happen like for good reasons, or it could just be like some weird technical thing. This, for example, I told my friend, what this MALAT1 is? It's like really, and MALAT1 shows up in like every single cell analysis. It's got to be some kind of technical issue with maybe how it's mapping or I don't know what. So I was like, or no, you think it, yes, what is it? Matan knows, Evgeny knows, yeah.
We're not sure exactly what it is. But it's an RNA binding protein.
Yeah.
That what we're actually capturing appears to be TALAM1, not MALAT1, for whatever reason. And we find that it's correlated, low abundance of MALAT1 seems to be correlated with just bad quality cells. And one hypothesis that it's a very rapidly degraded RNA species.
So I have exactly the opposite, where I have high MALAT1 with small cells or low quality. So yeah, who knows? Anyway, whatever. The point is, you have to look at all these with some suspicion, right? This could be a really cool iron-related gene. Maybe there's a really cool, awesome process, or it could just be like, whatever. Yes?
Talk to people in my lab about MALAT1.
Okay.
It's one of the most important non-coding RNAs.
I believe that, but I don't have any idea if it's meaningful here, right? Right. So the point is, before I do anything with these, I need somebody to do some experiments and tell me if this is meaningful or not. So these also got left out, but that doesn't mean they.
CRIP1.
Yes.
When you say it's mostly that gene, what does that mean? The topic is really just CRIP1 or not?
No, yeah, the gene. So I had a talk in 1 second, if I get to it, about how I chose genes, but the topic is kind of distinguished by that gene. It's most distinguished by that gene. And instead of being kind of distinguished by a lot of genes, that's like a very strong signal in it. So actually, let me come to that really quick. I just want to say, choosing the number of topics, I have no idea what to tell you. I can tell you, this is the plot I put in a paper. This is actually how I did it. So the plot I put in the paper is, because of the software that I used, output a likelihood. So I was able to actually compute AIC and BIC, and usually this isn't particularly helpful. I've seen lots of examples where these aren't particularly helpful, but basically I was like, okay, here's some values for different numbers of topics. I can see my BIC is like creeping back up here. It looks kind of low here, and here, AIC is like plummeting, and then maybe it's getting lower. I don't know, whatever. It's like, this is maybe 15 is a reasonable choice. I can make that argument. Here's why I really chose it. What I really chose is that I know that type 2, that this side of this blob represents type 2 activation. And I know that type 3 activation is what's causing my pathology. I know that these have to come from somewhere, and we were kind of thinking about a connection between these two, right? So if I see a topic that unites them, I am like sold on that. I want to follow that. And what I found is that in other renditions of this, possibly just for stochastic reasons, these got a little bit more separated, right? Because you could also represent this as two topics that were kind of like the way they get grouped doesn't necessarily have to do that. And I was like, oh, this is something I really want to follow on, and I have a reason based on my science of why I think this might actually be real. It's not real by itself; it's just suggestive. Okay, let me.
And there is no similar topic in the 10-topic model?
What did you say?
There is no similar topic to this one, like when you have only 10 topics?
So in my very, very rudimentary testing, I found it was a little bit less strong. Like it'd be more here and less there, or more there and less there. This was just one where I was like, it looked like really convincing, and I was like, that's the one I want to follow up on. And in fact, my collaborator was like, if you don't follow up on that, I'm not, like at some point you don't have that many, you have some directive from outside as well. So here, just want to really quick show how these kind of compare with the clustering, right? So one thing that's really nice is, so here I'm coloring by cluster those cells that had very high topic 11 weight. Does that make sense? So I've grayed out everything that wasn't high for topic 11, and I'm just coloring these by these cluster colors. And I'm showing kind of a gene that's characteristic of topic 11, and you can see, okay, this topic bridges these clusters. It would be harder to find with a differential expression analysis that was cluster-based. Doesn't mean that you couldn't. It's just nice that you kind of can see that relationship. Whereas here, I've got this topic 13 that bridges these, and I can see SRGN is one of these genes that kind of is important in topic 13, and I can see it kind of it drops this cluster. So that means the clustering was picking up on probably interesting real signal. It's just nice that I can find these topics that bridge across different clusters or divide up what might by other means kind of look like one thing. So for me, that's like useful. It's not probably surprising for you. Like it is very distinct from clustering. Okay, so I'm gonna run out of time if I don't speed up a little bit. I don't wanna like rush too much, but I just wanna say I picked 6 topics. I picked them based primarily on the fact that they were related to immune activation or immune repression, and that was the process I was studying. And they distinguished in some way. So here, these are the ECDFs, and these are colored by the different conditions, and in most cases, they distinguish some kind of activated state from some repressive state. And so I just thought, okay, this is useful to at least follow up on these. It doesn't mean other ones wouldn't be interesting. So in terms of how I use the genes, it's just worth a mention that the software that I used, it was coded by Matt Stevens, who did the admixture estimation. He was like on the original paper, and you know, a postdoc, I assume. And they have this function where they basically do almost kind of an enrichment. So they use KL divergence to pick the genes that are most distinguishing of a topic. And that is kind of like throwing out the common words, right? So if you have a gene that's it's important in a topic, but it's highly weighted in several of them, it won't get selected by this. It's good in some ways. I now realize it's also problematic in other ways because it might not have that high a weight. So I think some combination, and I would redo this a little differently, but that's what these represent. So these represent the score that you get from as a sort of a, you can think of it like differential expression or something like that.
So in the more recent literature, a lot of the algorithms that have polynomial time guarantees for these kinds of models, actually, those guarantees exist only if you have these kind of anchor words that are for example, the usual example is given the word bunt, and you're doing New York Times sections, right? If you see the word bunt, B-U-N-T, you can be pretty sure this article has some sports, and probably baseball. Right? So I think intuitively, that's exactly what you're saying, right? These are genes that when you see these genes,
right, and they may not have the highest weight just because, right, exactly. So that's exactly right. They may not have the highest weight because bunt may not appear as often as bat in a baseball story, but it's very specific. That's exactly the example, So these are kind of very specific. This is kind of this repressive topic that's very much in the naive. These are, some of these I didn't know exactly how to interpret. This is a lot about antigen presentation. If I say that, my collaborator gets upset because we don't actually know that it's doing antigen presentation. It could be related to other processes, but whatever, they're cool, they're real, they're cohesive. Then these are activated. These are topics more associated with activation. So again, here, this is this topic that looks like classical ILC2 activation. We see IL-5. It's the only topic in which you see IL-5, and that's a real thing. You see IL-13, which is also classic, and you see a bunch of other ones. And actually, IL-13 isn't represented here because it's kind of everywhere. And it's more sort of specific to topic 15 because that's like where you don't expect it as much. Okay, let's just go through what I did. Okay, so here's just a proof of principle of how you might use this. There are probably other ways, different ways. But what I did is I said, okay, it looks like there are these kind of transitions that might be present because I have this kind of continuous blob. I'd like to understand how these topics relate to each other. So maybe I could try diffusion map analysis with them. But I want to use the ones that I think are actually relevant. I want to try to get rid of the ones that just have to do with cell size and things like that. Try to focus on some topics that I can interpret and understand. So I basically chose cells that were highly weighted for those 6 topics I just showed you. I got rid of any cells that were highly weighted for the kind of cell quality type topics. And then I chose the top 50 genes for each of those topics and excluded anything that was like in the top 5 for these topics, basically. And the top 50 was by this measure. Okay, and then I made a diffusion map. So what's interesting about the diffusion map is that I have kind of this triangular plane at the bottom, which is where all of the cells that are not in the induced mice, they don't have psoriasis, they weren't treated, all of those lie here. So do some of the ones that were induced, but basically everything that wasn't treated lies in this triangular plane. It's pretty well spread out by the wild type. It's a little bit more enriched in one corner for the ones without T cells. And then I get this direction that looks like this. So this is like DC2 and DC3, and then DC1 captures this kind of direction where I see induction kind of moving that way. It's suggestive. Doesn't mean anything. You can make diffusion maps of lots of stuff that don't really exist. So, but one thing that's cool is that in this triangle, you see a pretty continuous dense filling out of the triangle, which means to me that, for example, cells are, they're really doing a lot of repressive topic AD-ness in this corner. They're really doing a lot of like antigen presentation, ILC2-ness in this corner. They're doing a lot of I don't know what in this corner, but they're kind of filling out all these different weights, right? Like, so we see that there really is a kind of continuum. You could expect, this might look really different otherwise. This is colored in by topic. And then I have, these activation topics, they kind of, some of them like this, the 113, starts down here and moves up like that. And one of the things I wanted to do was just kind of focus on this trajectory a little bit, because that's the process that is maybe potentially most revealing for us. So I then focused even more on this specific topics that kind of outline that trajectory. So here, this is a different diffusion map, and you can see you get like this kind of axis where you've got all the untreated cells, and then moving this way, you come into the induced, you get the separation between wild type and rag, and basically you see topic 8 is here, topic 13 is here, and topic 3 is here. So that really suggests, because you basically, there are some cells that go from here to there, but most of them go this way, and that suggests possibly that to get to this activation state, which we know is pathological, you have to go through this state. Now, it could be that it's just some technical aspect of my analysis, but what's cool, so this is a heat map where I just ordered according to that DC1, and you can see that you get what looks a lot like a kind of continuous span of gene expression. These are genes associated with the different topics, and you can kind of order them in this way where you see like these don't all come on at the same time. I mean, of course, there's lots of caveats, but it makes it seem like this isn't a really discrete process going on. So one thing I want to say is that the part of what I love about working in Aviv's lab is that if you're computational and you get to do a bunch of stuff, it doesn't just end with like, here's a bunch of predictions, which all might be trash. So some aspects of this actually got validated. So my collaborator had worked for a long time to build this mouse, which can report if IL-5, which is this very type 2 specific cytokine, ever gets turned on. So it's a fate map. So basically, it expresses this yellow color if ever the cells ever express IL-5. It expresses red if they express currently IL-5. And then he crossed that with a mouse that reports for these type 3 cytokines 17 and 22 with green and blue. So basically, you can see current expression of any of these cytokines, and you can see historical expression of IL-5. And then we also did kind of a re-challenge to try to get more of these cells, because this is actually really hard. Basically, if you give IL-23 again after the cells calm back down, then you get an even bigger response. After the mice calm back down, you get an even bigger response, which makes it easier to actually measure what's going on. So basically, what you see is, after the re-challenge with IL-23, you get more IL-5. This is current, you get more 17A, you get more 22. We know that's happening, so we're picking up on signal, that's good. And what's cool is that you also see that a good percentage of these cells that express type 3 cytokines used to express IL-5. They don't currently, but it means that actually that trajectory is a real thing, right? They went through, they turned on IL-5 at some point, maybe it was already there, maybe it got induced, and then they turned it off and they became ILC3-like, and they start expressing type 3 cytokines, not all of them, but this isn't a perfect reporter either, so we don't really know exactly what the percentage is. And that's kind of important if you think about treating this process or breaking it up, because you want to know that those cells didn't just like appear or come from somewhere else. You want to know that they could be. You might need to interrupt, say, type 2 activation, even when you're treating with this thing that's creating a type 3 pathology. And then I guess I didn't show it here, but the other thing is that we also validated, we see this co-expression of IL-13, which is another type 2 cytokine, with 17 and 22. That might not mean anything for you, but what it means to me is that these two cytokines that are both considered type 2 are regulated differently, right? One of them gets turned on and turned off when something becomes type 3, and there's this trajectory, and the other one can actually just coexist with type 3, and so maybe type 2 and type 3, it's like this mixed state that exists that isn't really so discrete. So we have both of those components. We have a continuous state that's going through a process we didn't know existed, and we also have this mixed state. Okay, so I think I'm at the end. I just want to, let's see, we got dense continuums, continuous states in ILCs. We get a new kind of mixed subset. Also, I mean, the whole idea of this, there being actually a repressive kind of state, that was also new, not really shown. and we can do a focused downstream analysis. I think in general, the real power with LDA is like what you do with your results afterwards. And that takes a lot of experimentation in my view. It's not obvious what to do, and then we actually experimentally demonstrated that really quick. Acknowledgements. Piotr Bielecki in the Flavel lab did all of the wet lab experiments except for Monica, who used to be in Aviv's lab. She's at a company now, and she did the single-cell genomics experiments here. And Aviv and Richard are both awesome. And so is everybody else, including Caroline, who's sitting there and helped me with some of the analysis. So that's all. Thank you.